Good morning. So I'm going to speak about the in vitro mutant models, so the mutated cell lines that my colleague Bertrand Collet developed at the lab. Bertrand is not here today, but he was interested in making those cell lines to understand better the structure of the type 1 interferon pathway in salmonids. I'm not going to speak very long about the type 1 interferon pathway. We had already several speakers commenting this. I would like simply to mention now that as in all innate immune pathway, this is based on a sensing step, an initial sensing step by different families of receptors, inducing signaling and the expression, the induction of many effectors. In the case of the antiviral response in vertebrates, this sensing leads to the expression of a master cytokine, which is a type 1 interferon. There can be several genes very closely related, and then this would induce through the YAG-STAT pathway a number of ISGs. What is important to remember here is that those ISGs, so interferon-stimulated genes, they can be effectors, they can also be regulators, and they very often have a role in a positive feedback loop inducing again type 1 interferon. So the structure of this pathway is rather complex. And it's important to remember that it's very redundant, which is nothing surprising because it has to be very robust in front of viruses that have many subversion strategies. So the loss of function experiments we made aimed actually at trying to understand better the role of key genes inside this pathway, and also more practically to try to generate cell lines in which viruses could be produced with a better efficiency, because obviously this is a limiting step for vaccine production. So how to do a loss of function experiment for this kind of genes? A long time ago, we tried with Jean-Pierre Levrault in zebrafish to do it with knockdown using morpholino, which is a very classical strategy, and we failed for almost all ISGs, probably because they are very strongly induced and the morpholino do not really succeed to stop the expression. Of course, the knockout in vivo provides a very good alternative, but it's very often rather long and expensive to do, even in zebrafish. And if the gene is essential, it can play many roles, it's lethal. So it does not always provide a successful model. So Bertrand actually wanted to set up a protocol to make knockout in cell lines, which may actually seem very simple, especially for people coming from the mammalian world. But in fish models, it's not so obvious. First, because the fish cell lines are not easy to transfect. It might be a very silly practical problem, but it's a real one. And also because cloning is a bottleneck. Because, of course, when you have mutated your cell, you get a mix of mutated and non-mutated cells. And it's necessary to clone cells. And actually very few cell lines accept to grow when they are as one cell in a cell culture well. So Bertrand finally succeeded to set up a protocol. We have a Chinook salmon line that is permanently expressing GFP. We can transfect guide RNA and Cas9 to make the mutation of the gene of interest and of the GFP in the same time. And we get a bulk population. We can have a first check to check that the expected mutated site is really mutated in the chromatogram. And then for the cloning, we get, after limit dilution, some clones that are GFP positive and some clones that are not GFP positive, like this one. And those clones are expected to have been mutated by the guide for GFP, and most probably also mutated for the other guide that should have been transfected in the same time. And it works rather well, so we have a good screening system to relatively quickly isolate the relevant clones that we can then validate after cloning for the presence of the mutation on the two haplotypes. I'm now going to go quickly through two examples of what has been done within this project. The first gene is the cytoplasmic helicase MDA5. So MDA5 is a sensor. It actually binds double-strand RNA, which is an important step in the replication of RNA, most RNA viruses. And this is one of the pathways leading to the expression of type 1 interferon and then ISG, then the antiviral states in the cell. So we used one guide. We could verify that in one of the cell lines we had a deletion of 2 nucleotides in the expected site, so it was really mutated. And in another control cell, we had just a substitution that had no effect. And we used it for control. So the C1 is a mutated and the C2 is a control. In this case, when we infected cells with the alpha virus, the salmon alpha virus, we got almost 1 log, well, approximately 1 log, being optimistic, of replication, of additional replication, while the control cell line was behaving as a parental cell line, and which was completely consistent for the expression of one of the type 1 interferon, the A3. We had a normal, very, very high induction of this gene in the control line and a very reduced induction in the mutated line. Then we tried to check many other viruses, and I'm showing here what happens with the SHRV, that is a rhabdovirus, one of these many rhabdoviruses infected fish cells. And we can see here that, again, the mutated cell line has an impaired induction of interferon phi1 in IRF3, that is another key transcription factor of the type 1 interferon pathway. MX, that is a very classical effector, is also a little bit reduced, but not so much. This is one of the gene of the pathway that is not much affected by the mutation in MDA5. But what was also interesting to see was that we had actually no effect on the replication of this virus. It was rather similar between the mutated and the non-mutated cell line. And this is something we have actually seen in many of the mutated cell lines we developed in this project, which was not especially what we wanted, because we would like to have cell lines producing a lot of viruses, but which was not really unexpected because it's so redundant. So just to summarize what we have seen about MDA5, we got a mutated cell line. We have a quite clear effect on the induction of the genes of the interferon pathway. No drastic effect on the cytopathic effect that I have not shown, but it was true for many viruses. And a slight gain in this case for the replication with one RNA virus, but not for all the other viruses we tested. Second example is RSAD2, that is also known as viperin. That is an effector genes, typical ISGs, almost all the time induced after viral infection or interferon treatment. And the main mechanism known for this gene is actually the conversion of CTP into DDH-CTP, so it's an analog of a base that actually impairs the replication of viruses in many models, not all, but many cases. So we developed a mutant for this. We used two different guide RNAs, and we validated them in vitro before in vivo. And the reason was that this was in a cyprinid cell line. It was actually one of the tasks we had to do in this project to try to extend the protocol to other species. And so we did that in a well-known and well-used cyprinid cell line, the EPC line. So we wanted to know if the guides were working, and they were working. And then we used exactly the same protocol as well as the one I described before, and we got mutants. In that case, the viperin being very well conserved structurally, we could use a monoclonal antibody to show by Western blot that in the wild type, after induction with poly-IC, you have expression, but this expression disappears in the two cell lines, in the two mutant cell lines we could select. Okay, so we analyzed the transcriptome of these cell lines, and I'm showing here what we have seen for one of them. In this axis is the difference between the knockout and the control at the steady state, and here in this axis is the difference between the knockout and the control after stimulation with interferon. So we have the corresponding population of genes that are affected by the mutation in one or in the other context. And briefly, what I would like to mention here is that if we consider what happens after induction with interferon, with recombinant interferon, in the mutant and in the control, we see that it's almost, there is almost no effect of the mutation. So the viperin appears really like an effector, and in this case, there is no obvious effect of the viperin on the induction of the other ISGs. Another thing that was interesting was that even if the EPC cell is not spatially close to an osteoclast, we could find at the steady state a clear effect on the mutation on genes involved in the ossification pathway. And that was funny because this gene was actually described in the 90s initially in rats as a gene involved in the activation of osteoclasts. So there is really clearly something affected at this level also in our mutant cells. And this illustrates also that the genes we mutate to learn about the interferon pathway can obviously have other functions that are more or less or maybe totally disconnected. And so just to conclude, we have generated a number of mutant cell lines in mainly with the CHSE, with the Chinook cell, Chinook salmon cell. So for the PKR, the helicase rigi, the two transcription factors that are very essential to the induction of type 1 interferon, RS7 and 3, MX, that is the myxovirus induced gene, that is an important effector, and MDA5, and the one I mentioned in the cyprinid cell lines. We are currently working on the phenotype. This is not very original. What is very important is that the effect of the mutation is very variable with different viruses. And the main lesson is that the system is so redundant that for the moment, we have very few cases in which we have an effect on the viral replication efficiency. We almost all the time have a very clear effect on the induction of the pathway, but apparently there are other mechanisms keeping virus down, which is, of course, quite interesting. And the last slide is to thank all people who were involved in the project, Bertrand being really the main person in the development of these mutants.